Okay, so picking up where we left off last class, I just started introducing to you membrane proteins, and we started talking a little bit about the functions of membrane proteins. We're going to go into more detail about some of these as we progress through the different chapters. They will play a role in each of the different chapters in a different way. But as hopefully remember from Chapter 2, proteins make up a huge portion of the biomass of a cell. They are hugely important to cell function, and just like they are elsewhere in the cell, they're hugely important to membrane function. Cell membrane proteins will act in many different roles. One of them we talked about a little bit in last class, and we'll talk a little bit more about in the next chapter when we start looking at how different substances cross cell membranes. They act as transport proteins, which will allow passage of ions and other substances through that non-polar region. So anything that's polar, not going to be able to get through the center of the cell membrane without going through a transport protein. And because of this, transport proteins are going to be able to regulate the passage of ions and other polar substances through cell membranes. So by opening and closing these transport proteins, you can determine what goes through the cell and what doesn't. Something we'll see more of in our chapters on photosynthesis and cell respiration are membrane proteins that act as enzymes. There's going to be a very important one called ATP synthase we're going to spend some time talking about. Enzymes are substances, or typically proteins, that facilitate chemical reactions, and we'll learn more about that in Chapter 4. But enzymes can be bound to the cell membrane and play a role to facilitate a chemical reaction well embedded in the cell membrane, and we'll see some of those. A big job of cell membrane proteins is recognition or cell identification. So one of the things we'll see in a few slides, we're going to see something called the extracellular matrix, which is stuff that's put on the outside of a cell. The stuff that's on the outside of your cells is unique to you. So proteins in certain cells within your body will interact with the stuff that's on the outside of your cells to determine if it's coming into contact with you or it's coming into contact with something that's not you. So if you have something in your body, like a bacteria that shouldn't be there, your body will be able to recognize it as foreign and attack it. So that's called an immune response. You'll be able to have an immune response by recognizing foreign invaders. Unfortunately, if that process breaks down, you can recognize yourself as foreign and start attacking your own cells. That's called an autoimmune response, and that can have bad consequences as well. But normal processes, normal immune behavior requires recognition, being able to recognize yourself from other things, from non-self. Adhesion proteins, so attachment. So like your muscle cells will attach to your bone cells. That's mediated by proteins. The attachment between neighboring cells within your stomach, within your blood vessels, those are all proteins that mediate those, that allow for a lot of functions that would not be possible otherwise. We wouldn't be able to digest food without attachment proteins. We wouldn't be able to move our arms and legs without attachment proteins. And receptor proteins, response to stimuli. Your cells need to be able to respond to things that are going on outside of them. We're going to talk more about this when we get into that part of the lecture handouts called 3A, when we start looking at cell signaling. We're going to learn more about receptor proteins and how your cells can have an internal response to something that's going on the outside of them, and that's again mediated by these membrane proteins. So here's just some pictures of those different kinds of proteins. So cellular transport, again, we'll go into this in more detail in the next chapter. Enzymatic activity, we'll go into more detail on that when we get to photosynthesis and respiration. Signal transduction, that will be something we'll talk more about at the end of Chapter 3. Cell recognition, that was what I just talked about with your immune response. So your immune cells, or your white blood cells, will be able to recognize stuff that's on the outside of your own cells and should know that it's you that they're touching and not something foreign. Intercellular joining, this is what happens in your muscles attached to your bones, your muscles attached to your muscles, your stomach cells attached to your stomach cells. We'll learn some more about intercellular junctions as we go along. And finally here, attachment to the cytoskeleton or extracellular matrix. This is something I'm going to go into right now. Your proteins that are embedded in the membrane will be attached to components that are on the inside of the cell that we're going to talk about on Wednesday called the cytoskeleton. And on the outside of your cell, they're attached to things called the extracellular matrix, which we're going to go into right now. So, in animal cells, on the cell surface, there's a structure called an extracellular matrix. So it's not just smooth surface, there's actually stuff sticking out on the outside of your cells. And most of that is carbohydrate bound to membrane proteins. So the purple is a membrane protein, the green here is a carbohydrate. So they're carbohydrates which are bound on the outside of your cell. On the inside of these membrane proteins is going to be internal cellular structures. And this is how your cell kind of advertises itself as being what it is. This is also a way for your cell to interact with things outside of it. And we're going to talk more about this when we get to cell signaling. A lot of that has to do with what's going on the outside of the cell. Different cells will have different structures on their surface, and you have different structures than the person sitting next to you on your cell surface, and allows your cells to act differently, but also allows you to recognize yourself versus foreign cells by what structures are actually there. So carbohydrates on the outside of the cell are attached to proteins embedded in the membrane, and those proteins are then themselves attached to structures on the inside. So you can probably see already how if something comes into contact with this carbohydrate on the outside, it could pass a signal into the inside of the cell and cause a reaction to occur. And we'll talk more about this at the end of this chapter when we look more at cell signaling. So that's what the outside generally looks like of an animal cell. Plant cells have a very distinctive feature on the outside of their cell membranes called the cell wall. So here's a plant cell membrane. They do have cell membranes as well. Outside of a plant cell membrane, they secrete carbohydrates in large quantities called cellulose. So they're going to secrete cellulose outside of their cell membrane to create a large protective structure called cell wall. And the cell wall serves several purposes. One, it's going to maintain the shape of the cell, and we'll talk about this later on today. We'll look at how that happens. But it also prevents water loss, and that's one of the big kind of challenges for plants. Plants can't get out of the sun if they're in the sun. They can't go find water really easily like an animal might be able to do. So they need to be able to keep themselves from losing water when they're in a kind of harsh condition. And one of the things that does that is the cell wall. The cell wall is often kind of this waxy barrier that prevents evaporation. Okay, so we'll talk more about this later on today, and then kind of in the next chapter as well when we look at how plant cells respond to water loss and things like that. Okay, but cell wall in plants, extracellular matrix in animals. Okay, the rest of today, we're going to spend talking about the various structures that are found commonly in the inside of a cell. So eukaryotic cells, unlike prokaryotes, have what are called organelles. And we're going to spend the rest of the day kind of listing off the various organelles that are found in typical eukaryotic cells and their functions. And you really should have a good idea of the major organelles and their most common functions. I'm going to end with kind of review question that you should be able to answer pretty easily. And then we'll watch a little video that animates all this and shows you some interactions between these different organelles. So prokaryotic cells, like bacteria, they're just one big room. They have no internal cell membranes. They have DNA. They have a cell membrane and a cell wall, and that's about it. Eukaryotic cells have lots of internal membranes separating the internal structures into organelles. And what organelles do is they each have different specialized functions that carry out the work of the cells. So when we go through and talk about these different organelles, we'll learn what their major role is, and they are optimized for doing that role. And because of this, eukaryotic cells are very efficient at performing a lot of these tasks, like protein synthesis, like protein excretion, protein modification, photosynthesis, cellular respiration. Those processes all have their own location where they can occur. Most of the structures that we're going to talk about today are part of something called the endomembrane system, which is a network of interconnected organelles, which means they're connected either by they're physically touching each other, or they're connected by things called transport vesicles, which I'll show you as we go along. Transport vesicles are just little sacs of membrane that can transport things from one location to another. So most of the internal structures are part of this endomembrane system, which allows your cell to transport material from the nucleus all the way out to the cell surface, to the cell membrane. So the parts that are considered part of the endomembrane system starts just outside the nucleus, something called the nuclear envelope. Includes the endoplasmic reticulum, the Golgi apparatus, specialized sacs such as lysosomes and vacuoles, as well as the cell membrane. So this is really what we're going to talk about today. And then in addition to this, we'll talk about some other specialized structures that perform their own tasks that are really separate from this system. So there's a really kind of nice kind of walkthrough in your book of the endomembrane system, and it follows a certain production of proteins. It follows milk protein production that starts at the nucleus, and it works its way all the way out to the cell surface for excretion of milk proteins to milk ducts for something like a pig feeding a piglet. So it kind of puts in a real-world example of how this endomembrane system is functioning. So I'd encourage you to read about that. This is your endomembrane system. Here's your nucleus, here's your nuclear envelope, your ER, your Golgi, and the cell membrane. And there are also vacuoles and lysosomes that we'll talk about as well. So we're going to start at the nucleus and work our way out and talk about these different structures. I'll give you some images of them and talk about their major role within the cell and what function they play a role in. So the first thing is the nucleus. Nucleus, in most cells, the nucleus is the most prominent structure. If you have done the microscope lab, and you have looked at Elodea, you may not have seen a nucleus. That's because the nucleus is clear unless you stain it. So unless you put a stain on that cell, you won't be able to actually see it. But it is there, and you probably would have been able to see it if you did stain for it. Its role is that it contains the cell's genetic material, DNA, and its role is protecting the genetic material, protecting the DNA. So the nucleus is surrounded by what's called the nuclear envelope, which is a double membrane. It's 2 layers of plasma membrane that separates the nucleus from the cytoplasm. So cytoplasm is the fluid within the cell, and it contains nuclear pores, which are holes in the envelope, in the nuclear envelope, that allow material to come and go. So proteins to enter and leave. It allows RNA to transport across the membrane, allows things like that. But it also regulates what goes in and out of the nucleus. So DNA is the information for how to make proteins in your cells. You don't want it to be destroyed. You want it very well protected, so it actually is surrounded by 2 membranes to very well protect it. You may, when you're looking at the nucleus, and you'll probably see this when you get to the cell division lab later on this semester, and you're looking at slides of onion cells, a lot of them you'll see within the nucleus a dense spot or a darker dot within them, something like that. That is something called the nucleolus. And the nucleolus is the region where ribosomes assemble, and we're going to talk about what ribosomes are next. So that structure, the ribosome, is actually assembled within the nucleus in the nucleolus, and that's the darker spot, and then it gets transported out into the rest of the cell. So nucleus, location where genetic material DNA is housed. Here's what the nucleus looks like. So here is a nuclear pore. So there's several nuclear pores on the envelope, and they regulate what goes in and out. You can see a little purple ring around them are proteins that can open and close the nuclear pore and determine what goes in. So here is a transmission electron micrograph image that's been turned the same color as the cartoon there. You can see there's a darker spot here, which is a nucleolus. So nucleolus, that's where ribosomes are assembled. And then the DNA is spread throughout here. DNA will condense, it will expand throughout the life of the cell. We'll talk more about that when we get into the DNA chapters later on. And you can see the pores kind of in this image as well. Okay, so what's assembled in the nucleolus is something called a ribosome. Ribosomes are very important for cells because they are responsible for protein synthesis. And we're going to learn a lot more about these later on when we talk about protein synthesis in one of the later chapters in this semester. But for now, just know ribosomes are responsible for protein synthesis. Once they are produced in the nucleus, they are sent out into the fluid outside the nucleus, and they're found in one of two locations. They can be in the cytoplasm or cytosol. I apologize, I use that word kind of interchangeably. Really, for the most part, means the same thing. Cytoplasm and cytosol is just a fluid that's out in the cell, in which case the protein would be made in the cytoplasm, or they can be bound to a membrane, and the two membranes you'll find ribosomes bound to are the endoplasmic reticulum or the nuclear envelope, in which case the protein would be made into one of those two structures. So if you're going to make a protein that's going to function inside the nucleus, it makes sense to make it on the nuclear envelope and actually have the protein fold and be produced inside of the nucleus. We're going to see that proteins made in the endoplasmic reticulum are going to be proteins that you're going to excrete at some point. So ribosomes, responsible for protein synthesis, found in multiple locations. I'm going to get to this. Okay. So one of the locations where you'll find lots and lots of ribosomes is bound to the endoplasmic reticulum. The endoplasmic reticulum, I'll draw these as I do them. The ER is a membranous sac, a series of membranous sacs or tubules that are attached to the nuclear envelope. So they're found just outside the nuclear envelope. So something like this. They actually take up a huge amount of room. It's really the majority of the membrane that's inside the cell is the endoplasmic reticulum. It takes up a huge amount of space. We're just drawing a little bit of it here. There are two regions to the ER. One is called the rough ER, and one is called the smooth ER. The rough ER has a bunch of dots all over it, and what those dots are ribosomes. And we know that ribosomes produce proteins, so which part of the ER is going to have protein production? The rough or the smooth? The rough, right? All right, it's pretty straightforward, right? Ribosomes produce proteins, ribosomes are found in the rough ER, so the rough ER is the site of production of proteins, and these are going to be proteins that will be secreted, and we'll see why that is as we talk about where products of the ER go. The smooth ER has a function of its own. The smooth ER, which lacks ribosomes, one of its main functions is that it is the site of lipid synthesis, and one of the lipids that it synthesizes a lot of is phospholipid, which is membrane lipid. So the ER actually is a location where membrane is produced. It's a membrane factory. The membrane can then come off of the ER as a little vesicle, and let's say you wanted your cell to grow, you might send that packet of membrane to the cell surface to increase the size of the cell membrane on the outside of the cell, right? So these vesicles, these transport vesicles, can leave the smooth ER as well, or sorry, the rough ER as well, and when they leave the rough ER, they're often carrying protein with them. And, well, first let me show you this image. So here's an image of the endoplasmic reticulum. Here's a cartoon. You can see the dots. These are ribosomes on the ER. Here's your smooth ER. Here's a vesicle that's leaving the ER. Vesicles could have proteins in them if they come off the rough ER. They could just be vesicles of just membrane if they're coming off the smooth ER. Here's again a actual image of the ER and the rough and the smooth, and you can actually see ribosomes when you look at them through TEM image. Okay, so this vesicle that might be leaving the ER containing proteins, where it might be headed is to another membranous structure called the Golgi apparatus. And the Golgi, which could be here, Golgi is just another membrane sac kind of structure. The Golgi's role is modification, sorting, and repackaging of products from the ER. So those proteins that came off the ER going to the Golgi, before they are going to be secreted by the cell or before they're going to be sent to another structure within the cell like the mitochondria or the chloroplast, they may need some modifications. Maybe parts of them need to be cut off to modify what was made by the ER. They may need to be folded in a certain way, and folding might be encouraged within the Golgi. They may need to have things stuck onto them. So the Golgi will take carbohydrates and stick them to the protein, making something called a glycoprotein. Or they might take carbohydrates and stick them to lipids that were made in the smooth ER, and that'll be called a glycolipid. So you can make these different products. They're going to be combinations of different biomolecules that we've talked about, and maybe you're making a protein that's going to be embedded in the cell membrane and be part of the extracellular matrix. So you stick a carbohydrate onto a protein, and then that protein will be sent to the cell surface to be embedded in the cell membrane. That modification is going to happen in the Golgi. The Golgi also sorts and repackages molecules into transport vesicles. So it acts like a shipping warehouse. It determines where these different products are going to go. Is it going to send a vesicle to the cell surface, or is it going to send a vesicle somewhere else in the cell? Some of the other structures we're going to talk about, that's determined by the Golgi. The Golgi will repackage them into vesicles headed in different directions. Okay. So modifying, sorting, and repackaging. So here's what the Golgi looks like. They often say there's a receiving end and there's a shipping end. So transport vesicles coming off the ER come into the receiving side. Those products get modified, get repackaged, they get sent out the other side, and they may head to the cell surface, they may head elsewhere in the cell. And in a cross-section of the cell, it just looks like a bunch of little sacs and transport vesicles. You can see them coming in on this side. Okay, one of the locations that the cell could send these vesicles is our next structure. So let's say one of these proteins was mistakenly made. Either it's not needed or there was an error in making it. So it's not a functional protein, and that's kind of determined prior to it being excreted by the cell that this protein has an error, it doesn't function properly. What your cell could do is it could send it to a structure called a lysosome. Lysosomes are vesicles that have enzymes in them called hydrolytic enzymes. And if you remember the reaction called a hydrolysis reaction we talked about in Chapter 2, hydrolysis breaks down biomolecules. So hydrolytic enzymes are enzymes that encourage hydrolysis to occur. So they encourage the breakdown of biomolecules. So lysosomes will dismantle and recycle food particles, so things from outside the cell. They'll also recycle the cell's own organelles, something called autophagy. So they're able to break down their own structures. So in this image, it shows a damaged mitochondria being broken down by a lysosome. So the lysosome merges with the mitochondria and breaks it down. Here's a lysosome that's taking food particles from outside the cell and breaking them down and partially digesting them. So your cell is really good at recycling components, so it's not going to let anything go to waste. Anything that it's manufactured that's damaged or not functioning properly is made up of biomolecules. It can break it down into the monomer of that biomolecule and reuse it. So it could take an old protein, break it down to amino acids. It could take a carbohydrate, break it down into a simple sugar, and it can then reuse that some other way. Either it can extract energy from it, it can use it to make a new protein, something like that. So lysosomes have hydrolytic enzymes. They will recycle or break down molecules. So you might send it to a lysosome. There are other sort of sacs within cells that are really useful for specific tasks. One of them is most commonly found in plants. There are smaller versions of this in animal cells, but we mostly when we refer to a vacuole, the second one here, we're referring to a vacuole in a plant cell. Vacuoles are simply fluid-filled sacs. In plants, the large central vacuole serves a similar function to lysosomes, so it will be a repository for waste. It'll be a location to break down and recycle components. But one of the main roles of the central vacuole in a plant is to actually allow the plant to have some structure to its body. So it exerts pressure on the cell membrane and the cell wall, consequently, to allow plants to stand upright. So plants do not have skeletons. So they have, you could refer to it as a hydrostatic skeleton. So they use water pressure to create stiffness to the plant. So here you've got a plant. You can see that plant cell, and you can see that the center of the plant cell is mostly empty. That's the central vacuole, and it's just full of fluid. And it's pushing out on the cell wall. You might see this in the Elodea cells. When you look at it in lab, you'll see that most of the chloroplasts are around the edge of the cell. And that's, again, because of the central vacuole pushing out from the center. So when a plant is properly hydrated, the vacuole is going to push out and make the plant stand up. But if you don't water your plants for a while, they'll start to droop. And the reason for that is that the vacuole shrinks in size, shrinks away from the cell wall, and the cell wall can begin to collapse on itself, and the plant will start flopping over. This is why in grocery stores, they're constantly misting their produce with water to keep those cells hydrated, to keep them plump so that they sell better, because you're not going to buy wilted lettuce. You're probably going to buy crisp lettuce. So you want to keep that stuff from losing water. Right. So exerts pressure, allowing plants to stand upright. Okay, and then the final one of these three, peroxisomes. This is not drastically different than a lysosome. It's a little more specialized. Peroxisomes are really good at disposing of toxic substances. They get their name because one of the main toxic substances that they detoxify is hydrogen peroxide. So hydrogen peroxide is a byproduct of some chemical reactions. It'll be broken down in the peroxisome and made less toxic to the cell. So similar to lysosomes, they will break things down. But typically, your toxins. So that's the endomembrane system. There's a few other components of the cell that I want to talk about briefly, and then I'm going to show you a video that kind of shows you a lot of these structures. So two structures that we're going to spend entire chapters on include chloroplasts and mitochondria. So I'm going to start with chloroplasts. Chloroplasts are responsible for converting energy from sunlight into sugars, into organic compounds like glucose, sugar, carbohydrates. They will capture light energy with the help of pigment molecules. Pigment molecules such as chlorophyll. Chlorophyll gives most plants their green color. It's just a pigment molecule that aids in absorbing light energy. We're going to talk a lot more about this when we get to the chapter on photosynthesis. So this is what a chloroplast looks like. Kind of a neat thing about chloroplasts, is that they contain their own DNA. So there's DNA housed in the nucleus, but there's also DNA housed in chloroplasts. And the reason for this is how chloroplasts came about. And if you take 1011, or if you take maybe a botany class, you might learn a lot more about this, as you'll start talking about the evolution of plants and how plants came about originally. But the theory is, the most accepted theory now is called endosymbiont theory. And what that is that a photosynthetic organism, like a bacteria that was photosynthetic, was engulfed or eaten by a eukaryote, so a eukaryote that got its energy by feeding off of these photosynthetic prokaryotes long time ago. And it was able to then use that bacteria to create energy for itself. So it no longer had to feed. It could feed off of the products of the photosynthetic bacteria. And in exchange for that, it housed the bacteria and protected it from being eaten by anything else. So there is actually there are organisms that are alive today that support this hypothesis, this theory. There is a sea slug, which I'd encourage you to look up because it's one of the coolest things. There's a sea slug that it feeds only once in its life. And it feeds on an alga called Vaucheria. And Vaucheria is kind of unique in that it has no internal cell membranes. It's just a big tube. It's called siphonous alga. It's just a big tube filled with chloroplasts and nuclei. And so the sea slug, just after it's born, will bore a hole in one of these algas and suck out everything from the center of that cell, including the chloroplasts. It'll then take the chloroplasts, and the chloroplast will be placed into the cells on its back. And for the rest of its life, which is only a few months long, it will actually use the chloroplast that it stole from that alga to generate sugar, which I think is crazy that something could do that, right? It'd be really neat if we could do that. If we just, you eat some lettuce and then you can walk around the rest of the day and you feel full because you're generating sugar by sitting in the sun, right? It would be great. But that's what it does. It's a process which has a really cool name. It's called kleptoplasty because it steals chloroplasts, so kleptoplasty. But it's evidence that this exists, that they can steal, that there are organisms that can steal chloroplasts and then use them. So then kind of the extension of this is that instead of having to feed every time there is a new slug born, maybe it started to be able to have the chloroplast reproduce at the same time that the slug reproduced. And that would be kind of the next step in the evolution of a plant. And these were all single-celled things at the time. This is a much more complex thing. It probably wouldn't happen nowadays, but with an organism like that, it would happen more readily with a single-celled organism that just had the chloroplast in it. But that's the idea of how this came about and why there's still DNA. There's still ribosomes. It produces its own proteins. It has a lot of unique things that allow chloroplast to function kind of independently from the rest of the plant cell. Very similar thing has occurred in pretty much all prokaryotic cells to give us mitochondria. Mitochondria are organelles that are specialized for a set of reactions called cellular respiration, and again, we'll have an entire chapter on this. Cellular respiration allows us to extract energy from food. These are found in prokaryotic cells, they're found in practically all eukaryotic cells. So they're found in animals, they're found in plants, they're found in fungi, they're found in pretty much any organism that is a eukaryote. It's going to have mitochondria in it. They also contain their own DNA. So we also think that they originated from this endosymbiotic relationship between a bacteria and a eukaryote, and the bacteria provided ATP for the eukaryote, and the eukaryote provided a housing for the bacteria, and eventually they evolved to reproduce together. So they again have their own DNA, their own ribosomes, they produce their own proteins, but we're going to see in the chapter on cellular respiration that the shape and the structure of the mitochondria make them really, really good at producing energy, and we wouldn't be able to survive the way that we do without them existing. So we'll learn a lot more about those. Okay, so here is a summary question, and hopefully you should be able to answer this at this point. So which of these 5 things is one of the roles of the Golgi apparatus? And we're going to see if we can pick out what the other ones are referring to. All right, last few responses. Awesome. Okay, good job. 100%. So, all said sorting and repackaging, you would be correct. So sorting and repackaging proteins into transport vesicles is a role of the Golgi. So which structure is responsible for manufacturing proteins? That's the location, right? But what's the structure that does the manufacturing? That's the ribosome. So the ribosome does that. And you're right, it does happen in the ER. That's kind of the factory for it. But the actual machinery is the ribosome. So how about storing genetic information, like DNA? DNA. Right. And genetic information is DNA. But where is that? That's in the nucleus, right? The nucleus. Recycling old organelles and housing digestive enzymes. That would be the lysosome, right? Manufacturing lipids. Yep, so that would be the smooth part of the ER, right? Okay, good. So hopefully you recognize these locations pretty quickly. That means you did a pretty good job of paying attention today and staying awake, right? That's basically the big take home from today was being able to pick out these different structures and their location. So I'm just going to do kind of an overview, a few overview slides, and then I'm going to show you a little video that will show you a lot of the structures that we talked about today, but also a lot of the structures we're going to talk about on Wednesday as well. So, okay, so here's a typical animal cell. Animal cells, you can see they're dominated with the nucleus and the ER here. You can see the rough ER, the smooth ER doesn't, well, this would be the smooth ER in here. Here's the Golgi. The Golgi can then send structures to the cell surface to excrete proteins. It could send things to lysosome that might break them down. Here's your mitochondria, which is your site of cellular respiration. So you can pretty much see all those different things, and you should be able to identify a lot of these things. These parts, the cytoskeleton, we're going to talk about that on Wednesday. We're going to get into what those structures are. So that's a typical animal cell. Typical plant cell has pretty much all those structures that the animal cell had, with a few extra. So it still has the nucleus, still has endoplasmic reticulum, it has rough and smooth ER, it has Golgi apparatus, it has mitochondria here, it has a cell membrane right here, but it also has a cell wall on the outside of the cell membrane. It has chloroplasts, which are sites of photosynthesis, and it has this large central vacuole. So the central vacuole takes up most of the room in a typical plant cell, whereas the nucleus in the ER kind of takes up the most of the room in a typical animal cell. The vacuole will push everything towards the edges, and you'll probably see that in your lab. But plant cells do have pretty much everything that animal cells have, plus a few more things. Central vacuole, well, that's what we kind of went into. It performs a lot of roles like the lysosome. So it's a location for waste to go to. It can break down some biomolecules, but it also works to exert pressure on the cell wall to give the plant cell structure. So by the vacuole pushing out on everything, it's making the cell wall rigid and allowing the plant to stand upright. So you'll actually play with that in your lab. You'll do, I think it's lab 6 when you're doing osmosis, you'll actually put salt water on a plant cell, and you'll see that the central vacuole basically disappears. It shrinks away, and the cell membrane pulls away from the cell wall. And when that happens, the plant cell collapses on itself, and the plant is now no longer standing upright. So that would be a wilted plant. So don't water your plants with salt water. That would not work out well. We'll talk about why that is in the next chapter, really. Okay, so those are both typical kind of generic plant and animal cells. Most cells are not, I guess most cells kind of have those structures, but they may have them in different quantities, and they have a lot of specialized features here. So upper left here, this is a muscle cell. Muscle cells have lots and lots of mitochondria. So whereas a typical animal cell might only have a few mitochondria, muscle cells are packed with mitochondria because they need to generate lots of energy in order to function like a muscle cell. Nerve cells here have really unique shapes, and we'll talk about how they hold those unique shapes on Wednesday when we talk about other structures inside the cell that give a cell its shape. So that's why nerve cells are going to have these really crazy shapes, and that allows them to do their role of transmitting signals across long distances within your body. Here are two plant cells. Here is a cell from, I don't know if I know, this is a leaf cell, so it'd be a lot like the Eladia cell that you see. And here is a cell from an onion, from the white part of like a white onion. So you'll notice that there are lots of green chloroplasts here, but there are no chloroplasts here. So even though this is a plant cell, yes, it has a cell wall, yes, it has a large central vacuole, but it doesn't have any chloroplasts. Why would this onion cell not have chloroplasts? Yeah, so it grows underground, so there'd be no reason for it to photosynthesize, so it wouldn't want to expend energy creating chloroplasts. If you looked at green onion, you looked at the leaf of an onion, it'd have tons of chloroplasts, but that one doesn't have it, whereas a leaf cell is going to have an excessive amount of chloroplasts. So onion root tips, even tree roots, you're not going to see chloroplasts, but tree leaves, you're going to see tons of chloroplasts. And that's why you have some regions of the plant that are green, they have lots of chloroplasts, some regions that don't. So a lot of those structures are variable. The number of the different structures are variable as well within a cell. Okay. So now what I'm going to do for the rest of the class, I'm going to show you a video. This is a video called The Inner Life of a Cell, and it is done by Harvard University. It's a kind of an animation of the internal components, and I'm going to narrate kind of over top of it. But it's a really cool kind of introduction to cellular components, and it'll show you a lot of the stuff we saw today, as well as there'll be kind of a preview for stuff we'll see on Wednesday as well. So this is a few years old now, but it's still a great video. Okay. So it's starting just in a blood vessel, and it's going to zoom in on this cell. So you can see here's the extracellular matrix of an animal cell. Got lots of carbohydrates. They allow the cell to communicate and attach to neighboring cells. Here's the outside of the cell membrane. This is the phospholipids, and there's proteins embedded in those phospholipids. You can even see lipid rafts. These are things we kind of talked about the other day. So now it's going to start going into the cell. You're going to see a lot of these cytoskeletal elements we're going to talk about on Wednesday. So this is cytoskeleton. That is what supports the shape of the cell, and we'll talk about how that works on Wednesday. One of the neatest things is how the cell can rearrange the cytoskeleton. So it will rearrange structural components of the cytoskeleton. It'll also rearrange these things called microtubules, which you'll see in a minute. And microtubules act like cellular highways within the cell. So these vesicles that are moving between the ER and the Golgi and the cell surface, they don't just float there randomly. They're actually carried by these motor proteins. So this is a transport vesicle. This is a motor protein, and it literally walks along a microtubule. So energy is burned as the motor protein moves, and that's how things get around inside your cell. So your cell has a lot of control over where things go. Here's your nucleus. Here's some RNA coming out of the nucleus. It's going to meet up with a ribosome and start making a protein. So this would be a free ribosome, making it just in the cytoplasm. So here's your protein coming out of a ribosome. But ribosomes can also make proteins when bound to a membrane, and you're going to see a protein being made on the rough ER. So here's the rough ER. There's a protein being made by a ribosome into the rough ER. And again, this protein can be packaged in a vesicle. The vesicle doesn't just float, it's carried by a motor protein along a microtubule. I think that's the coolest thing. And it might carry it to this structure. You recognize this? It's your Golgi. Yep. So it's going to be sorted and repackaged. Some of it might be sent to the cell surface and excreted here, and this is how you get surface proteins. This is also how your cells can excrete things into the bloodstream to be passed around the rest of your body. So here again, we're on the outside, cell surface, and by rearranging those structural elements within the inside of the cell, you can actually cause movement of the cell on the outside, and we'll talk more about that on Wednesday. So, pretty cool video. This will be on Canvas, so you will be able to watch it again. There's tons of versions of this on YouTube. There's even some that have different narration where they go into a lot more detail about the different structures, if you'd like. And then this group, the BioVisions group at Harvard, has lots of other videos as well, so I would encourage you to look at their website. They have a really good one on mitochondria. Okay, so that's it. We'll continue from here on Wednesday.